Okay, so any question about previous one, previous chapter? Anything you didn't understand? That was the basic thing. Okay, so from today we are going to start the basic things of genetics. The first thing is that what is chromosome? Because we'll talk about everything about DNA and RNA, and DNA is located inside the chromosome. So we need to know about the structure of chromosome, how DNA is packed within the chromosome, and how DNA is transferred from one cell to another cell. That is the process of cell division and reproduction. Okay, so these are the main things we'll talk about, and how the genes are transferred here. So the first basic thing is that what is prokaryote and what is eukaryote?
Prokaryote is very simple and it has like a nucleus with a single.
What does it mean, this term, this prokaryote and eukaryote? What is the term means? In science, when we are naming something, it is not like that only the name of the scientist or something like that. Sometimes we are doing that, but it has some kind of meaning most of the time. What does it mean? This just this word, prokaryote, eukaryote. What does it mean by karyote? Carry on. Actually, the actual word is carry on. It is a Greek word. Carry on. Carrion means nucleus. Okay? Carrion means nucleus. So that's why when we are talking about the cell division, we called it as karyokinesis. There are two processes. One is called karyokinesis, one is called cytokinesis. Karyokinesis means nuclear division. So karyon means nucleus. Pro means primitive. Prokaryote means primitive nucleus. Eu means new. Eukaryote means new nucleus. Okay? So these are two terms. If you remember these two words, then you can understand the things. Now, prokaryote means primitive nucleus. Eukaryote means modern nucleus or advanced nucleus. So what is the difference between these two? What is the difference between prokaryote and eukaryote? First thing is the difference between their nuclei. Difference between their nuclei. So one is the primitive nuclei, another is the modern nuclei. So what is the difference between primitive and modern? How can I differentiate between these two? How can I differentiate between these?
Are there no structures in the DNA or in the nucleus?
Structures means what?
Like they're not separate, they're all mixed up together. There's no nucleus.
Make it more specific. So actual thing is that nucleus, that is the DNA, the genetic material. DNA, the genetic material is packaged within the nucleus in eukaryote, but it is not within the nucleus in prokaryote. Okay. Generally, this structure in prokaryote is called, this DNA is more or less free, or something, sometimes it is jumbled up. It is called a nucleoid rather than nucleus. It is called nucleoid. Okay. So this is one thing. Another thing, another difference between prokaryote and eukaryote. This is the nuclear difference. Another difference about their DNA. Just you need to remember all those things throughout your whole life. The question is that what is the difference between their DNA structure or DNA packaging? DNA packaging. So they have the DNA, it is open. Here there is the DNA which is inside the nucleus. That is one thing. But they have some kind of thing there in their DNA packaging. DNA is coiled up generally inside the nucleus or outside the nucleus, whatever it is. It is coiled up. But there is something there, the difference between their packaging, this coiled packaging. Histone protein. In eukaryote, histone protein is there. So DNA and histone protein together can coiled up to make it as chromosome, whether there is no histone protein in eukaryote, in prokaryote. Okay. So in eukaryote, I will talk about this one later. There is a histone octamer. This is called histone octamer. So that means octa means 8. So there are 8 molecules of histone proteins there. They are together, and this is called the histone octamer. In this histone octamer, DNA is coiled up with 1.5 times here. Okay, each and every time. And this beaded structure is called chromomere. This structure is called chromomere. So chromomere is present in eukaryote but not in prokaryote. And the presence of histone protein is very, very important, particularly for the DNA replication and protein synthesis. Okay, that is one. Another thing, another difference between eukaryote and prokaryote, not in nucleus, but as a whole together. What? Mitochondria, every cell needs mitochondria because mitochondria is the powerhouse of cell. Okay.
That's what I was gonna say.
Okay.
You carry.
No membrane-bound organelle is in prokaryote. And in eukaryotic cell, you will find membrane-bound organelles. Okay. So these are 3 main differences between prokaryote and eukaryote. In prokaryotic organism, what is an example of prokaryotic organism? Bacteria. And eukaryote? Plants. We are eukaryote also. Plant and animal cells. There is another term, it's called archaea. What is archaea? Have you gone through animal classification before, animal or plant classification?
Archaea or archaea is like ancient.
Archaea is not actually the oldest form. The bacteria is called the oldest form. Archaea is a typical type of bacterial, that's prokaryotic structure, which is very, very specialized. Suppose in the hot spring, very hot spring, like 100 degrees centigrade, there you will find some kind of prokaryotes which are habituated or adapted with that environment. Those are called archaea. And if you think about the evolutionary trend, then the archaea is more close to eukaryote rather than bacteria. Bacteria is a little bit far from the eukaryotes, but archaea is more close to eukaryotes. Okay, so these are 2 examples of prokaryote: the bacteria and archaea. Okay, so these are things. Nucleus in prokaryotic cells generally absent. The true nucleus, the whole thing we call nucleoid. Eukaryotic cells, the nucleus is present. Cell diameter, generally prokaryotes are very small, eukaryotes are larger. Genome, usually 1 circular DNA. Generally, in case of prokaryotes or bacteria or archaea, you will find the circular DNA. But circular DNA is also present in some eukaryotes, but generally in eukaryotic DNA, it is linear. Okay, this is the difference. Circular DNA versus linear. Amount of DNA relatively very small because there is no such chromosome, only the DNA is present, so it is very small. While in case of eukaryotes, we have a lot of chromosomes. Each and every chromosome contains 1 DNA molecule, so it is relatively large. This is the explanation why it is large. And membrane-bound organelle is generally not present in prokaryotes and present in eukaryotes. So these are the differences between these 2. Size is not really very much that is distinct, but sometimes we need to talk about the size also. The size of the prokaryote is very small and eukaryote is very large. Okay, couple of things I just need to talk in general. Sometimes I can skip some slides because already I have talked about that one or it is not needed. But I will put all the slides in your canvas so you can have all the slides there. And if you think that I am going too fast about that one, just check me there that you cannot understand that part. So I will repeat that. Okay. So this is the structure of actually histone. So there is one histone octamer, and each and every histone octamer, the DNA is coiled twice, almost 1.5 to twice, 2 times. Okay. Now, what is virus?
It's like a capsid that just contains,
What is life? I
it's
have a
not
question. What is life? What do you mean by life? We are in the life science. What is life?
reproduce, they can reproduce.
They can reproduce and they can respire, they can have metabolism, they have distinct structure, okay, and they can reproduce, and also they can respond to stimuli. So if we have cold, we need to wear the sweaters. If it is too hot, we need to wear the shorts. So we are responding to the stimulus, okay, the temperature. So these are the differences between life and non-living or non-living things. What is virus? Virus can be crystallized. You can crystallize sugar. You can crystallize not salt. Yeah, salt crystallization is also available. Rock salt and other forms of salt. You can crystallize those things. Any physical matter. You can crystallize virus also. Okay, and virus contains DNA material, genetic material, either DNA or RNA. There are some viri called the DNA virus. There are some virus called the RNA virus. Okay. So what is virus now? Is it a living thing or not living?
Not-
How many are saying that non-living? Okay. How many are saying living? Only two. This is in between living and non-living things. This is the only form between living and non-living things. We cannot differentiate whether the virus is a living thing or virus is not. You can crystallize virus, you can keep it for a long time, there is no problem, but their DNA material, DNA is very, very actually stable. Because that is one example is there. Can you tell me that example? The DNA is very stable. You can find the DNA. Any movie, from a movie. From a movie.
Outbreak,
What?
outbreak, outbreak, movie outbreak.
Jurassic Park.
Jurassic Park. Okay, one mosquito took the blood from the T-Rex. That blood was inside the mosquito, and that T-Rex cell, that DNA was inside the mosquito, and it was crystallized in a, it was fossilized in some form. They took out that mosquito, they took out that DNA, and from that DNA they created the T-Rex. Okay, so science is there. Definitely, the science is there, and not only that. In the next one, like the third one, the third, not third, the fourth one, that is the Jurassic World, the first Jurassic World. There is one dinosaur that is called Indominus Rex, right? Irex. There is another science there. There they couldn't find the some DNA part matching with that dinosaur. They took the T-Rex DNA, they took the other DNA. Finally, they discovered that this is from the Velociraptor. That part. They took the frog DNA and they matched all those. So every science is there. If you saw the movie, if you see that one, you will find that finally they discovered the some part, the missing part of the DNA they took from another DNA, and that DNA was Velociraptor. That's why that Indominus Rex was communicating with the Velociraptors. Okay, so science is everywhere. even in the movie. So this is the thing. The virus generally contains one capsule, and this capsule is generally the physical matters. Something is solid sugar structure, carbohydrate or protein structure is there, mainly the protein structure, and there is one capsule there, and within this capsule they contain the genetic material. And main function of the virus is that they need to transfer this genetic material into the host. Now the question is that what is host and what is parasite? Host and parasite. Yeah.
The parasite can't live without the host, so the parasite has to have
That's
a host to
another thing. But at
survive.
first,
And it-
suppose there is one mosquito and me. Who is host? Who is parasite?
What about host and mosquitoes?
Mosquito is the parasite. So virus generally is a kind of parasite. Parasite means who is transforming or who is depending on other body for their survival, but creating harm into that host. That is called the host and parasite. Generally, virus is a kind of parasite. Okay, it is transforming its genetic material into host genetic material, and that is the example of AIDS. HIV, it is a retrovirus, and these virus contain some kind of genetic material, DNA, and that DNA is incorporated into human host DNA, and that's why it is replicating there. Okay. Okay. Now the prokaryotic cell replication or cell division, it is called cell reproduction, but I can say cell division because their reproduction, as they are unicellular, that means their cell division means their reproduction. They are reproducing new form of different organisms. They have, it is very simple, just simply binary fission. This is called binary fission. What is that? In this process, DNA is replicated into two, and the whole cytoplasm is replicated into two. This is as simple as that. Okay. Now there are a couple of terms here, which is called the origin of replication. Do you know what is origin of replication? What is DNA replication? In a very simple term. Suppose I am not a scientist, I am not from science. If I ask you that you are learning biology, can you tell me what is DNA replication? How can you?
It's making an identical copy of the DNA.
Yeah, it is making identical copy of DNA from one DNA. Okay, that is as simple as that. And what is origin of replication?
The very beginning of
In a particular part of the DNA from where the replication starts. Particular part of the DNA from where the replication starts. And that is called origin of replication. I will suggest you to write down all these terms. That is origin of replication, prokaryote, eukaryote, and virus, all these terms. It will help you to study all those things because the questions will be on these. Okay. And generally, in prokaryotic cell reproduction, as it is very simple, they don't need that much energy, that much all those things there. It is very rapid. Very rapid means if you culture a bacteria, within 1 hour you can find out 1 million bacteria there. It is in the log phase. It is called the log phase. It is going in the power rate. So 10 to the power 2, 10 to the power 4 in such kind of things. But so prokaryotic cell reproduction or cell division is very, very simple. But eukaryotic cell division, it is not simple. So what are the things there? We need to think about this couple of points. One is homologous pair. What is the structure of the chromosome? What is cell cycle? And what are the genetic consequences of the cell cycles? So if we need to study these things, then we can understand the actual cell division of eukaryote. Homologous pair. What does it mean? What is the word? Homologous. If
Same.
I divide the word like that, one is called homo and another is called logos. What does it mean? Homo.
Same.
Same. What is the opposite of this one? Better. Okay. Logos means, actually it is called logos, Greek word. Logos means the part. Okay. Or you can simply remember as locus or loci. That means homologous means same part. Okay. Or similar pattern, not similar, we call the same. Now, so homologous chromosome means, that means there will be two chromosomes which are identical in size. Not only that, their genetic configuration will be same. And in the same part. It may be recessive or dominant, but very actual. They are all identical. Okay, these are called homologous pair. Now, homologous pair, we have the diploid cells and haploid cells. What is diploid? What is haploid?
And diploid means there are two sets of chromosomes.
Di means two. Diploid means two sets of chromosomes, and haploid means half set of chromosome or one set of chromosome. Okay. So where can I find the diploid cells?
Are they somatic cells?
Somatic cells. What is somatic? What does it mean, somatic?
Anything that's not a sex cell?
Soma means body. So somatic cells means body cells. Okay. And haploid cells, well, what are the haploid cells? We can find it in gametes. So there are other two terms. One is called the somatic cells, another is called gametes. Gametes means sperm and ovum in case of animals. And in case of plants, it is spores. Okay, spores, ovum, and all those things. It is saying that one set of genetic information. What do you mean? That is a single chromosome or single DNA or some other things. What does it mean by one set of information? Suppose we are human, we have 23 pairs of chromosomes or 46 chromosomes. We are diploid. So that means we have just two chromosomes or we have a pair of chromosomes, we have 23 pairs of chromosomes. What does it mean by the diploid?
Means that we have a set of chromosomes with each parent. So each parent gave
Set of
us
chromosome means what? How many?
two.
Two. We are diploid, we have two sets of chromosomes. One set means how many chromosomes? 3? 23? What do you think? Others? Make it clear, make your concept clear, make your concept clear. Because these are the basic things what we require in our later classes. Okay? One set of chromosomes, that means total 23 chromosomes from either from father, either from mother. That is one set of chromosomes. So chromosome number 1 to 23 should be there. Okay? So that is called one set of chromosomes. Two sets of chromosomes or diploid means we must have 46 chromosomes, not only 46 chromosomes, 23 pairs. Because sometimes in case of chromosomal aberration, we can find that 46 chromosomes, but one chromosome is triploid. Okay, and another chromosome is missing. So we must have 23 pairs of chromosomes, and they should be homologous chromosomes. Okay, so there are so many terms. Right now I am making it a little bit complex. So homologous pair and 23 pairs of chromosomes in the full form, that is called a diploid. So this is diploid set our human chromosomes, and generally we can now color these chromosomes like that. And this is called chromosome painting. Okay. Later we will learn about this one, how to do the chromosome painting, and that is with the DNA probe. We need to add DNA probe with the fluorescent dye, and they can go to the particular distinct chromosome there. Suppose last day I was talking about that. How can we know that which DNA is from which chromosome? We know that this gene is from this chromosome, this gene is from that chromosome. We know that. But how can I know? Because when we are extracting the DNA, we will find the under the tube, we will find the white materials there, and that is DNA. And we don't know from which chromosome it is there. So we have to identify the gene, then we have to tag, we need to make the DNA probe, small DNA probe tagged with fluorescent dye. Then we can inject that one into the cell, and that will replicate with that particular DNA there and show us the fluorescence light. Okay, then we can identify, okay, this gene is there in this chromosome. And this is called the chromosome painting. Right now we can paint the chromosome in different colors to find out the homologous pairs. Okay. So diploid cells have two chromosomes, two sets of chromosomes, one set of chromosome, two pairs of homologous chromosomes. Which will be the answer? T. Two sets of chromosomes, not two pairs of homologous chromosomes. Two sets of chromosomes. That means 23 into 2. Clear? I need to look at your face that it is confident that I know that. Right now I know that, yeah. Okay, now we need to talk about the chromosome structure. Chromosome structure means chromosome is made up of generally two things. One is a long pair, like this kind of things. Here is a constricted part here, and then, but to draw this one, I am making this one as a round shape, but it is not actually the round shape. So there are two parts. One is the long part, and another is this constricted part. There are two things here. This whole thing, this long rod-like structure is called what? This rod-like structure, the long structure of DNA. What is the name of that one? What? Chromosome is altogether.
Is it a gene?
Gene is a part of chromosome, a part of DNA. But the whole rod-like structure, or whatever it is, thread-like structure, whole thread-like structure is called what?
Chromatid.
Except this part,
Chromatid.
chromatid. Okay, and what is this part? The constricted part, centromere. Now, if I ask you what is the difference between centromere and chromomere? What is the difference between centromere and chromomere?
Does the centromere just connects the single chromatid and the chromomere connects all of it?
Chromomere is formed with the histone protein and DNA coiled there. That is called the chromomere. Centromere is the part of chromosome where chromatids are attached. Okay. Now, based on this one, and there are other two things that is called the telomere and origin of replication. Okay. Now come to the structure. So this is centromere, and this whole long thing is called the chromatid. Okay. But our chromosome, our body system is very, very advanced. Even our science is not that much advanced. I am telling that one. Our body system is so much advanced. We have signal transduction system and everything. We are not advanced at all. That like that. So it has packaged. The chromosomes need to be protected from different kind of environmental factors. It is protected by some caps. Okay, two parts of the chromosome, two tips of the chromatid. It is protected by some kind of cap. This cap is called telomere. Tel means the tail, that is the end or tip of the chromosome, and there is some kind of cap that is called the telomere. Or generally, we cannot say the cap. The end part of DNA is called telomere. Okay, and this end part of DNA, it has some function. Generally, if we when we will go for the molecular biology part, we'll find that telomere is some kind of structure where the repetitive sequences are there. Repetitive sequences means something like A, T, T, C, G. Then you will find another A, T, T, C, G, and this repeats. So this one is one repeat. You can find hundreds or thousands of these repeats. The sequence is same. It is repeating all the times. This is called repetitive sequence. And in telomeric region, you will find this repetitive sequence a lot. Because during replication, each and every time it loses one sequence, at least. When we will go for the replication, you will find that one, how it is losing the sequence. And that means when it is losing one sequence, one repetitive sequence there, because it is not in, it has not been replicated by the process. So each and every time it is losing a sequence. So that means when chromosome is getting older and older and older, it is losing a lot of sequences there in the telomeric region. This is called aging. Okay. When we are aging, we are becoming aged. That means we are losing a lot of telomeric sequences there. Okay. Sometimes, if the telomeric sequences or something like that, the telomere is almost gone, or telomere is very, very slow, then there is a chance, very, very low amount of repetitive sequences. There are a couple of like 10, 20 or 100 sequences are there. Previously, it was 1,000 sequences. Then there is a chance of having cancer. Okay. So this is the relationship between aging and cancer. Generally, most of the cancer you will find during at the very later age. Okay. This is the sequence, and the telomere is responsible for that one, aging and cancer. And this is called aging of the chromosome. So that means during aging, the telomere becomes shortened. Okay, and that can lead to cancer. So this is the function of telomere, but telomere has different kind of replication process and different other functions too. And when we'll go for the molecular biology, we'll talk that one. So we have two things here. One is chromatid, one is centromere, and telomeric part. Now, in your book, you will find another part they called as the origin of replication. Generally, origin of replication is not like that because there are a lot of origins of replications in eukaryotic chromosome. But in prokaryotic chromosome, you will find suppose like 3-4 origins of replication. But that is there in your book. That's why I am telling that. But if you are asked that what are the parts of chromosome, two things, chromatid and centromere, and you can add the telomere. Okay, these are all. Now, this centromeric part, it has very important role. In the centromeric part, there is another protein is there here, a protein-like structure which can hold the spindle fibers. Okay, which can hold the spindle fibers. It is the attachment area for the spindle fibers with the chromosome. This part is called kinetochore. Okay. So centromere doesn't mean that it is kinetochore. There is some protein there which can hold the spindle fibers at the centromeric region of the chromosome, that is called kinetochore. Clear? Now, based on the position of centromere, the chromosomes can be divided into various groups. Okay. So first one is that There is a chromosome here, there is no centromere. Okay, there is no centromere at all. This is called eccentric chromosome. No centromere, eccentric chromosome. Okay, centromere can be of at the telomeric region, at the top, the telomeric region. If the centromere is there, it is called telocentric chromosome. During cell division, the spindle fiber is attached here. So in such case, you will find the chromosome eye-like structure. Okay? So that is called the telocentric chromosome. Now, if the centromere is just a little behind the telomeric part, just a little behind the telomeric part, then it is called acrocentric chromosome. In case of acrocentric chromosome, during metaphase, when the spindle fibers are attached, or during anaphase, you can find the chromosome like a J-like structure. Okay? It is simply J-like structure. You cannot find this part, but like this, J-like structure. Okay? And this small part of acrocentric chromosome is called rabbitium. It's called rabbitium. Okay? Generally, it is a common terminology, it is called the rabbitium. Now, if the centromere is between the middle part of the chromosome and a little bit lower than the acrocentric part, it is called submetacentric. Submetacentric. In case of submetacentric chromosome, you will find L-like structure. Okay. So during metaphase, we can find either I-like structure, J-like structure, or L-like structure. If it is L-like structure, L means the latter part is a little bit bigger than the J. So it is called submetacentric chromosome. If the centromere is truly between the middle of the chromatid, then it is called metacentric. And you will find the V-like structure. Okay, both arms are equal. In both these cases, one arm is long, one arm is short. But in case of J, that arm is very short. In case of L, that arm is a little bit longer. But in case of metacentric chromosome, you will find the V-like structure. Okay. Now the question is that for telocentric, acrocentric, submetacentric, and metacentric, you'll find there is a presence of centromere. But in case of eccentric, there is no centromere at all. So what is the function of centromere? I know that there is a kinetochore protein there in the centromere, which is helping the spindle fibers to attach into the chromosome. If there is no centromere at all, what will happen to that chromosome? Will it go for the cell division cycle or not? Go or not? No? If not, then what will happen to that? Generally, sometimes you can find it as a micronucleus in the chromosome, or it can be digested at all. It can be digested by the lysosome process because this is the extra material. Who can digest? The lysosome. Okay. So these are the 5 types of chromosome based on the position of their centromere. Okay, and just note it down, based on the position of their centromere. Okay, then another thing is that generally we know that in mitotic cycle, you have the idea of mitosis and meiosis before. So in the mitotic cycle, we know that chromosome is, DNA is replicated. Okay. So during interphase, how many chromatids can we find in a chromosome? During interphase. DNA is not replicated at all. How many chromosomes can we find? How many chromatids can we find in a chromosome?
One.
One. Okay, because DNA is not replicated. So this will be the structure of interphase chromosome. Okay, but it is more or less more threaded, like it is not coiled up in that much. During metaphase, we'll find the structure like this. Okay, two chromatids, two identical chromatids, because DNA is replicated there at that time. Actually, during prophase we can find that one. That is the two chromatids are there, but in metaphase it is coiled up. We can find it very clearly, and those forms you can find only in metaphase stage. Otherwise, you can find that metaphase and anaphase stage. The V-like structure, J-like structure. When we study the cells, we can take the metaphase place. We can arrest that one. We can arrest the metaphase plate. We can, so we used some kind of chemicals. It's called colchicine. If you inject the colchicine into the that is the cell, then it can destroy the all spindle fibers. Then the metaphase plate is arrested. Then we can study this kind of chromosomes. Okay, this is normal general technique. So we have two chromatids during our cell division process. Now, these two chromatids, if I consider these two together, what can I call one chromatid? The relationship between one chromatid with another one. Relationship between one chromatid of one chromosome with the one another chromatid of that chromosome. What is the relationship between these two? What term we can use? Sister chromatid. So these are called sister chromatids. Sister chromatids. Now there is one that chromosome, and here I have another chromosome. This chromatid and this chromatid. What is the term relationship between these two?
Like same or identical copies.
No, this is another chromosome. Suppose this is chromosome number 5 and this is chromosome number 9. So one chromatid of this chromosome number 9 with respect to the one chromatid of chromosome number 5, what is the relationship between these two? These are the basic terminologies, because later we will use these terms a lot. So we need to have the clear idea about these terms. What is sister chromatid? And that is called non-sister chromatids. If I say between exchange between non-sister chromatids, later we will use this term a lot during the DNA structural evolution or deletion duplication process, because there is some kind of things even in the meiosis. In this part, later part, we will talk about the exchange of chromosome materials between these sister chromatids or between non-sister chromatids of homologous pair. Okay, so you need to know the term. When it is non-sister chromatids, that means two chromatids from two different chromosomes. When it is sister chromatid, that means two chromatids from the same chromosome. Okay, this is the difference between these two. Clear? Now, each and every cell have a cycle process. So they are going into cycles. They have their life cycle. We all have life cycle. Every organism. Their life cycle consists of 3 stages. One is mainly called the interphase. What is interphase? Interphase is that when they perform all their cellular activities. All their cellular activities mean respiration, photosynthesis, if it is plant cell, metabolism, and fighting with the other things, that is immunosystem, everything. What are the function of the cells? Everything is there in the interphase stage. Okay. So from interphase, when they go for the divisional phase, that is in case of somatic cells, right now I am using all the terms, in case of somatic cells, when they are going for the divisional phase, that is called the mitotic phase or mitotic division. In case of germ cells, when they are going to this divisional phase, that is called meiotic phase. Okay. So interphase and then M phase. or divisional phase. Okay. And after this thing, this M phase, there is another part when the cytoplasm need to divide. Okay. When cytoplasm needs to divide, that is called the cytokinesis. So if that is cytokinesis, what is mitosis and meiosis? Cytoplasmic division, cell division, nuclear division, chromosomal division, what? What do you think? Cell division means cell contains both things. One is nucleus, another is cytoplasm. So what is mitosis and what is meiosis in terms of these two things? If you know that one, then you can find out the term cytokinesis. Or if you know the term cytokinesis, then you can tell that. What is mitosis actually mean? What kind of division? Separation of somatic cells, that is fine. So somatic cells division means mitotic division and germ cell division is meiotic division. That is fine. But what is actually mitosis? It is cytoplasmic division or nuclear division or chromosomal division. What do you think? Separation of cytoplasm is called cytokinesis. This is called karyokinesis. Karyon means nucleus. So nuclear division is actually two types. One is called mitosis, one is called meiosis. Okay. Cytokinesis is same for both type of cells. Cytokinesis, so what is the difference between karyokinesis and cytokinesis? Karyokinesis means division of nucleus. Only the division of nucleus. Then chromosomes will be separated. Then there will be another set of chromosome will be formed. Either it is diploid or it is haploid in case of mitosis or meiosis. That is the process of karyokinesis. Done. Mitosis and meiosis. Then after that one, cytoplasm is to divide. Cytoplasm is to divide means what is cytoplasm except the nucleus? That means all the cell organelles, Golgi bodies, plasmids, not plasmids, sorry, plastids, Golgi bodies, lysosomes, mitochondria, endoplasmic reticulum, all other things, they will be equally divided during the cytoplasmic process because cell needs all those things. Okay, so all the cell organelle division after the nuclear division, that is called cytokinesis. So this is the difference between these two. Cell division means mitosis means the division of nucleus in somatic cells. Meiosis means division of nucleus in germ cells. After the karyokinesis, there is always a cytokinesis to make the division fully adult or to make the new cells fully formed. Okay, so in this cell cycle, there are a couple of things. This part, this red part, it is the divisional phase. So we are talking about the somatic cells right now, and it is mitosis. So divisional phase is mitosis. Okay. So after the mitosis, there is cytokinesis. So two cells are formed. Right now, I am starting from that part. So this is a new cell. This new cell will go for the total interface stage. During interface, they need to perform all the cellular activities like respiration, metabolism, and all those things. Okay. This whole interfaces, so that means that yellow and this gray part, this yellow, and this gray part is the interface. So we can divide the whole interface stage into three main parts. One is G1 or gap 1 stage. Next, the S phase or synthesis phase. And later is G2 phase or gap 2 phase. So gap 1, synthesis, and gap 2. It is called gap 1 phase. So what does it mean by gap 1? Why did we name that the gap 1? Gap 1 means there is no synthesis. So what is synthesizing there during this process? Because all other activities is going on all the times because cell needs their energy, so the respiration is going on all the times. The metabolism is going on. They need the protein materials, carbohydrates, and other things for their cellular activities. So metabolism is going on all the times. So what is happening during this synthesis phase? What kind of synthesis is it? Any idea? That is the DNA replication phase. So DNA is replicated into this part, this synthesis phase. So what is gap 1? Gap 1 means there is no such synthesis phase, there is no such activity other than all other cellular activities. With respect to the cell division, there is no such activity. So what is the function there in gap 1 phase? The cell is formed, new cell is formed, it is very small at that time. It needs to attain its full size. Okay? So it needs the healthy food there. It needs to grow up. So that is the growing type, growing pattern that is happening into gap 1 phase. It needs to attain the full form of their body. So that is happening into the gap 1 phase. Now, one thing is that we have the skin cells. So each and every time, generally this whole cycle for the human, it is for 24 hours. And in 24 hours, the gap 1 phase is for like 7-8 hours. Interphase state is for another 12 to 14 hours. And gap 2 phase is for 5-6 hours. Okay, roughly like this. Okay, so in such case, I had the cells here yesterday, so I should have the double cells because it is already 24 hours. But why is it not? Yesterday I was something like 6 feet tall, so today I should be 12 feet tall. Theoretically, yeah, if it is 24 hours, theoretically that is true. If I am the student, I will ask the professor this question. Okay? So you are saying that it is taking 24 hours to divide, so why we are not like that? That is the question. This is the common thing we need to understand. Okay? So why not? Why not? Cell death, no. Cell is not dying in such a way. Cell has a particular cycle. Suppose sometimes it takes around 40 cell cycles after that cell dies. Or something like 100 cell cycles after that the cell dies. Cell dies, definitely, because each and every day we are having some dead cells from our body, from our skins. So, but they have a particular, not after just one cell cycle. It's something like 30-40 cell cycles or something like that. So why not? And another thing is that it is not growing every day, that's fine. But if we have a cart here, then those cells will grow to heal up that cart.
Are they smaller at the end themselves when they split up? They're smaller than the original.
Actually, it is called, yesterday, last time I told that one, they have some kind of checkpoints, whether it is required or not. They determine the thing, whether cell needs to divide or not. If there is a cart there, that means there is a requirement to divide the cells. If there is no cart there, we don't need to divide the cells. It is stopped. The cell division is stopped. Okay? That is called the checkpoint. That is called the checkpoint. G1 and synthesis phase, between these two phases, there is one checkpoint. Some molecules are there. There are a lot of molecules are there. Cyclin and other factors are there. They are determining whether this newly formed cell will go for the another cell division process or not. They will determine whether it will go for the another process of cell division or not. If there is a cart, then they will synthesize. They got that response. Okay, right now these cells, particularly in that area, those cells need to go for another division of phase, not the other cells. Okay, they will determine, that checkpoint will determine whether it will go for the division of phase or not. If they not go for the division of phase, they can be there. They can perform their all other activities, that is respiration, metabolism, everything is there, that is fine. If they need to go for the divisional phase, then they need to replicate the DNA and go for the division. So this point, if once they pass this point, they should go for division. Okay, there is no. They should go for division. That means there should be the DNA replication and all other things required for cell division. Clear? Now, our nerve cells generally, actually the brain neurons. Neurons, number of neurons we had during our birth, we still have that number of neurons. There is no extra neuron. Okay? It doesn't grow, it doesn't divide. So, what is happening there? And what is happening for our skins also? If there is no cut, they are staying in that phase. What is that? That is called G0 phase. Okay? So they can stay in that phase. They can stay in that phase. Our neurons are permanently in G0 phase. They will not go for the divisional phase anyway. Their structure is something like that. They don't have that kind of chemicals which can target them or we can tell them to go for the next divisional cycles. So that phase is called G0 phase. So our skin cells and everything is in G0 phase right now. It cannot cross the checkpoint. If it is required, then it will come out from the G0 phase and go for the synthesis phase. Clear? This part? Now, when they pass this checkpoint, G1 and S phase checkpoint, they will go for the synthesis phase. This synthesis phase means at that time, they need to divide into two. So we have 23 pairs of chromosomes. We need another 23 pairs of chromosomes. So DNA needs to be replicated. DNA needs to be replicated during that time. So main function is the DNA replication. So during this S phase, the DNA replication occurs. Now, then it comes to the G2 phase. When it comes to the G2 phase, it is preparing the cells for the cellular division. That means it needs to form the spindle fibers, so they need to accumulate the tubulin protein inside their body and so other things. They need the energies and other things. So that is preparing for the divisional process in the G2 phase. So there is no DNA synthesis there, only the preparation of the division. That is called gap 2 phase. There is no synthesis, that's why it is called gap. So gap 2 phase. When they have done all these things, then they check that whether I have the perfect number of DNAs, we have the perfect DNAs there, the DNA is replicated properly and everything, they are having that checkpoint. Another molecule, some other molecules here, all the investigators or invigilators are there, they are the molecules. Okay, they are checking that okay, DNA is replicated properly. Quality control. Okay, DNA is replicated properly, it is certified, then go for the division. If not certified, they will not go for the division. That is another checkpoint between G2 phase and M phase. Just before going to the mitotic cycle, there is another checkpoint who will determine whether the cell will go for the division or not. So if it happens that it cannot pass this quality control checkpoint, what will happen? That means DNA is already replicated, but the replication is not proper. In such case, the cell will be digested. And the lysosome will function that one. This cell, they give the signal, nucleus will give the signal, okay, this cell doesn't need to go for the division, it will be digested. Okay. Then it goes for the mitotic division. And in mitosis, we know there are generally 4 phases. Right now, some books call the 5 phases, including the pro-metaphase. But during this point, I am talking about that one. I will talk that point also. But during anaphase, during metaphase, actually, the spindle fibers are attached to the chromosomes. There is another checkpoint there, whether the spindle, just before the anaphase, there is another checkpoint, whether all the spindle fibers are attached to the chromosome or not, or whether all the chromosomes are attached to the spindle fibers or not, properly arranged or not. They check that part. If there is some disturbance there, that they are not properly arranged or they are not, all the chromosomes are not checked by the spindle fibers, they are something loosely arranged, they will stop the process. So that is another part. So it is a very, very programmed pathway, the cell division. Still that we are getting some kind of mutations, okay? So in such case, if the chromosomes are not attached to the spindle fiber. So during that time, there will be no cell division. Cell division will stop. Okay. So there are main three checkpoints are there. So we have two main stages. The first stage is called. So there are two main stages of the cell cycle. One is called divisional phase, and another is called interphase. Interphase can be divided into three phases: G1, S, and G2 phase. Between G1 and S, there is one checkpoint. Between S and G2, there is one checkpoint. And in divisional phase, we have, generally in your book you will find 5 phases, but generally we call it as prophase, metaphase, anaphase, and telophase. Okay, divisional phase at first we can divide into two. First is karyokinesis, and this is the part of karyokinesis, and cytokinesis. Okay, so divisional phase, divide that part into karyokinesis and cytokinesis. In karyokinesis, we have 4 things: prophase, metaphase, anaphase, telophase, and cytokinesis is another one. And this, in your book, you will find another stage here that is called prometaphase. Right now, in modern biology books, they are using this prometaphase, but generally we have 4 stages. So if you write the 4 stages, that is also true. If you write 5 stages, that is also true. Okay. Telophase. Telohs. Telophase. Telophase and anaphase. Now here is the checkpoint number 3. Okay. This is the whole skeleton of cell cycle. Now go to the mitosis. So mitosis means there are 2 steps. One is karyokinesis. So mitosis means the division of nucleus. And there is another one called cytokinesis. And there are 5 phases, or you can say 4 phases. So what is happening here? Okay. So this is the division of nucleus. So what we will talk here, we will talk about the nuclear division. So first thing is that the cell is there, and here is a nucleus. Including interphase, nucleus means this is called nuclear membrane. Okay. What is the structure of nuclear? This is nuclear membrane. Here you will find a sap-like structure, plasma structure. What is plasma? Is it a solid, liquid?
Semi-solid? Semi-solid?
Semi-solid. That is the stage between liquid and solid. That is called the plasma. So one plasma structure here, this is called nucleoplasm. Nucleoplasm or nuclear sap. Okay. You'll find another solid dense body here. This is called nucleolus. Okay, nucleolus. This is nuclear membrane, and you will find the thread-like structures during interphase. These thread-like structures, generally these are called chromosomes. Okay, but you will find a reticulated structure. because the chromosomes are overlapped with each other and it is very long. So you can find a reticulated structure. Reticulated means a network-like structure. Then it is called nuclear reticulum, or you can say chromosomes. Either nuclear reticulum or chromosomes. Nuclear reticulum term is only applicable during interphase stage. Okay, so this is our interphase nuclear structure. Okay. Now, what is the difference between plant cell and animal cell? What are the differences? 3-4 differences.
Chloroplast.
Chloroplast.
So,
Chloroplast is where?
it's
Where? Plant or animal?
Plant.
Plant. Then
so
cell wall in plant and I need some input from here, this side. This is very silent, except you. What are the differences between plant cells and animal cells? They said one, the nuclear, that is the cell wall and chloroplast. Other than that, lysosomes and vacuole, generally large vacuole in a plant cell, but it is not present in animal cell generally. But in animal cell, there will find a small vacuoles also.
Reproduction, plant cells use photosynthesis, how they reproduce.
That is the chloroplast. Not only chloroplast, actually there are plastids. Chloroplast, that is a green plastid. Chromoplast, different other colors. Leucoplast, that is a white color. Sometimes amyloplast or something like that. The flowers are different colors. That is for the presence of chromoplast. Okay. Endoplasmic reticulum is present in everywhere. That is in the plant cell and animals. Another main difference, which is really very, very important, particularly from the cell division process. Another organelle, which is present in animal cells, not present in plant cells. Ribosome is everywhere, even in prokaryote, because without ribosome, there is no protein synthesis. Ribosome is everywhere. Because ribosome is the factory of protein synthesis. Okay? And genes expressed through protein synthesis. So ribosome is everywhere. Another one. Another. What do you think?
right.
Cell membrane is there. Even in plant cell, after the cell wall, there is cell membrane. That's why we can see the plasmolysis. That is when it is detaching from the cell wall and it is in some form. Centrosome. Centrosome is present in animal cells but not present in plant cells. This is very, very important. Centrosomes plays very important role in cell division, in mitosis and meiosis. Okay. Generally, centrosome is present here in animal cells. Centrosome consists of two particles here, which are called centrioles. Okay. Two centrioles together in a capsulated structure is called centrosome. What is the function of centrosome? The centrosome is responsible for forming the spindle, the whole spindle. Okay. So during prophase, what is happening here? During interphase, we have this kind of structure. During prophase, what is happening there? The chromosomes coiled up. Chromosomes coiled up. Coiled up means they are, suppose if you coil a thread like that in this way, this way, this way, after sometimes you will find it is very thick. Okay, thicker structure. So chromosomes are thread-like structure, they are coiling up. So when they are coiling up, during prophase, we can find the nucleolus is still there, but chromosomes are, that time is visible. Like this. So how many chromatids can I find at this time in the prophase? 2 or 1? Per chromosome? 2 or 1? 1? 1 or 2?
Two.
2? Why 2? Why 1?
Three.
Because it already passed the synthesis phase. Synthesis phase means DNA replication is done there. If the DNA replication is done there, that means there should be 2 chromatids for each chromosome. Okay, it already passed the synthesis phase, DNA replication phase. That means we will find 2 chromatids for each chromosome. But still, the chromosomes are a little bit thread-like structured, and you cannot identify the chromosomes very well during the prophase stage. You will find during the prophase stage, you will find the nuclear membrane intact almost. Nuclear loss is always there, but the chromosomes are a little bit quiet. Okay. Now, what is happening after that? After that, the prometaphase, which is called the prometaphase, generally we called it as prophase also. During that time, the nuclear membrane becomes disintegrated. Okay, nuclear loss disappears. Centrosomes, these two centrioles, they are going apart from each other and forming the spindle fibers. Okay, chromosomes are more coiled up, so we can have a distinct structure of chromosome right now. It's not threaded. This is the structure of prometaphase. During metaphase, what is happening here? The spindle fiber is fully formed. So there are 2 types of spindle fibers. So these are the polar regions. 2 centrioles are in 2 polar regions. So if these are called the 2 polar regions, then this part is called equatorial region. Okay, poles and equator. This is called the equatorial region, the just middle of these 2 poles. Okay, so centrioles or centrosomes, right now we can call this because this is just single centrioles. Centrioles are in the polar region. Spindle fibers are formed here. Some spindle fibers are just connecting the one pole with another pole, 2 centrosomes. These spindle fibers who are connecting the 2 centrosome or 2 centrioles, there is no chromosome attached to it. This is called continuous fiber. That is called continuous fiber. Now, what is happening for the other chromosomes? Now, chromosomes are arranged just in the equatorial region. Their centromeres are there. All the centromeres are arranged in this equatorial region. Okay? Centromeres are connected with the spindle fibers. These are called spindle fibers. These are called continuous fibers, which is just between these 2, there is no chromosome. So in metaphase stage, the main thing is that chromosomes are all at the equatorial plane. Centromeres are arranged in a linearly here, and each and every chromosome is attached with a spindle fiber. This is the metaphase part. So there is no nucleus, nuclear membrane, no nucleolus is gone. Spindle is fully formed at this stage. Okay. Now what is happening after that? These spindle fibers, they are pushing each other. These continuous fibers, they are pushing, and these spindle fibers, they are pulling. So what is happening? If they push each other, these continuous fibers, these 2 centrioles will go further. Okay? Because it is pushing, so they will go further, and they are pulling. So for this pulling tension, these chromosomes can divide into the centromeric region. Okay? Tearing up the chromosomes. So during anaphase, what we will find here, cells are a little bit bigger in this stage. Two centromeres are little far apart. Now the chromosomes, now this part, the chromosomes are pulled towards the pole. Chromatids are separated here like this. Okay. During this stage, we'll find all the structures, J-like structure, L-like structure, V-like structure, then we can identify, okay, this is, because we can identify the actual centromere at during that stage. That's why I told that J, L, I, those shape structure, because of these. So this is called anaphase. And this movement of chromosomes from equatorial region to the polar region, this is called anaphase movement. Anaphase movement. So what is anaphase movement? Movement of chromosome towards the poles from equatorial region during anaphase of cell division, either is mitosis or meiosis, that is called the anaphase movement. Clear? Now what happens after that? So after this stage, the spindle fibers disintegrate. Okay? Spindle fibers disintegrate, centrosomes becomes having two centrioles again, and the chromosomes are here. Chromosomes are a little bit squared right now at that time. So they are extended. How many chromatids will be there during that? After the anaphase? During anaphase, how many chromatids you will find? The chromosome? One. Because until metaphase, we have the chromosome with two chromatids. During anaphase, the chromatids separate. So we'll have only one chromatids here. And there is no spindle fiber during this time. So we have the chromosomes with single chromatids. Nuclear membrane reappears. Nuclear membrane reappears. Nucleolus reappear here. During this phase, that is one cell having two nuclei, fully formed nuclei with equal number of chromosomes, this stage is called telophase. Okay. So if you, I think you did the biology 1081L lab, did you do onion root tip slice? Okay. In onion root tip slice, this stage, that is one cell having two nuclei, fully formed nuclei, this stage is called the telophase. Okay. Now what is happening here? Now there is cytoplasmic division. Telophase is done. So karyokinesis is done. Mitosis is done. Almost. Okay. Now after the mitosis, there is a process of cytokinesis. What will happen during this phase? All the cell organelles will replicate. Same number of Golgi bodies in both sides, same number of plastids in both sides, same number of centrosome in both sides. Centrosome is already there, centrioles are there, and mitochondria and all other materials, they will divide each other. We still don't know how they will divide, but they remain same. If one cell contains, the parental cell contains 107 mitochondria, the daughter cell, each and every daughter cell will contain 107 mitochondria. Okay. In case of animal, there is a process is called furrowing. So the cell membrane creates a furrowing process. It constricts around this equatorial region and finally reached together to form two different cells. Okay, this is simple cytokinesis. So what is happening with the plant cell during that process? First of all, there is no centriole in plant cell. Okay, if there is centriole here or centrosome here in animal cell, it is called astral spindle, because from the centrosome Actually, if this is centrosome, it will find the spindle fibers in the first way, this first process, just around the profile. It's something looks like a sun-like structure. The asters are formed. These are called asters. So in animal cell, the spindle is called astral spindle. In plant cell, there is no centrosome. That is called non-astral spindle. This is the difference between plant cell and animal cell. Astral spindle is formed in plant cell. Non-astral spindle is formed in plant cell. Astral is animal cell and non-astral is plant cell. But in case of plant cell, here we have the cell wall. Okay? Cell wall is a rigid structure. It cannot constrict like that. So what is happening there? Between these two nuclei, newly formed daughter nuclei, there is a formation of cell plate here. Lignin, protein, they are accumulating at the middle of these two nuclei. Then they will fully form the cell wall between these two. Okay? So this process is called cell plate formation. That newly formed cell membrane is called, the cell wall is called the cell plate. This cell plate extends towards the cell wall, that is the side of those cells, and they form the cell wall there. Okay? So in plant cell division, the cytokinesis is by cell plate formation. In animal cell division, the division is by furrowing. Okay, this is the difference between these two cell divisions. Clear? Okay, so I think I'll stop here. Only one thing is that
I'm not
just
sure.
remember, as I was talking about the cell cycle here, now this size of the cell is very small. It is not the actual size. That's why it needs to go for the G1 phase to attain the full size. Okay? So this is the function of G1 phase, to attain the full size. Okay.
My name is Madison. Nice to meet you. I just put it on YouTube. I did there's a live stream of it because I can't take notes as fast as most teachers talk. Yeah.